Hello all, my name is Liam Smith and I am a rising junior at the University of Massachusetts Amherst, majoring in biology and neuroscience. This summer I was allowed to participate in the University of Delaware Summer Institute, where I completed some research in the fields of genomics and bioinformatics. Today I will walk you through the pipeline of differential expression analysis. The first step in performing differential expression analysis is preparing data. We can prepare data that shows us the levels of expression of genes within the cell by performing RNA sequencing, which is a technique that allows us to isolate the mRNA transcripts that are present within a cell at any given time. This reveals the level of gene expression through seeing the level of the mRNA transcripts that relate to each gene. We're interested only in mRNA because these transcripts are those that code for proteins and will reveal levels of protein within the cell. We can perform RNA sequencing at multiple times throughout the lifespan of an organism to see how their gene expression profile changes with time or the development of a particular disease. Once mRNA is isolated from a cell sample, the quality of this mRNA is assessed using machinery, and the mRNA is then converted to DNA using reverse transcriptase if the quality is determined to be high enough to continue analysis. Only DNA can be sequenced in the sequencer due to the increased stability that DNA has over RNA. Once reverse transcription is performed, adapters are ligated to the DNA, and these adapters allow for the DNA to attach to the sequencer machinery so that PCR can be performed properly within the sequencer. PCR is performed within the sequencer to make sure that there are enough DNA molecules present for full sequencing of each DNA fragment. At this point, the sample is ready to be sequenced, and raw output data is converted to FASTQ format so that it can be computationally analyzed into readable and interpretable differential expression data. At this point, our data is fully prepared for differential expression analysis. This analysis entails comparing the expression levels between two groups of organisms to extrapolate potential effects of the differentially expressed genes between these two groups of organisms. At times, this is a very hypothetical and theoretical form of research because with only data on the differential expression of genes, only so much can be truly known about what's going on within the cells, and using these differentially expressed genes, we can make educated assumptions on how these genes are affecting the symptoms of a disease that is observed within an organism. With normalized RNA sequencing data, many analyses can be performed. In my case, I used the acquired differential expression data to perform pathway analysis by organizing the differentially expressed genes into clusters that were involved in certain pathways and trying to understand how these differentially expressed genes would affect the particular pathways that they were involved in. My research is focused around comparing differentially expressed genes in modern broiler chickens that are affected by wooden breast disease and those that are not. Wooden breast disease is a novel disease that affects the breast muscle of commercial broiler chickens, and it is characterized by muscle hardness and extreme growth of the breast muscle, and many lesions are present within this muscle, like lipid infiltration, fibrosis, hypoxia, and ultimately cell death. The cause of this disease sadly has yet to have been found, but using this differential expression analysis, we hope to learn a lot more about the possible causes of this disease and a lot more about the symptoms of the disease as a whole. An example of a gene that is differentially expressed between wooden breast affected chickens and their unaffected counterparts is the LYP-D6B gene. This gene is upregulated within wooden breast chickens, and it encodes for a protein modulator of excitatory receptors of the nervous system. The presence of this protein increases the receptor's sensitivity to their ligand, acetylcholine, and when acetylcholine binds to these receptors, muscle contractions are produced, and these receptors are responsible for the resting muscle tone. If LYPD6B is upregulated in these affected chickens, these receptors may show increased activity due to the heightened sensitivity that this protein causes them to have to their ligand. This overactivity is something that may contribute to the advanced muscle tone and hardness that is seen in affected chickens, along with immobility. That's an example of how we perform differential expression analysis using the knowledge we have about particular genes within the nervous system and other systems. I hope this video has helped you learn about differential expression analysis as a whole, and I appreciate you taking the time for watching.